Today on the deep dive, we're plunging into a world often overlooked when we think about animal health: fungal diseases. We're specifically focusing on two fascinating animal groups: Camelidae, that's your iconic camels, but also llamas and alpacas, and Cervidae, the elegant family encompassing various deer species like elk and moose. We typically hear about viruses or bacteria when animal illnesses come up, but fungi, they are truly ubiquitous everywhere, really. And the spectrum of issues they cause ranges from minor irritating skin conditions to absolutely life-threatening systemic illnesses in these large, majestic mammals. What's truly striking is how these microscopic fungal invaders, often overlooked, can profoundly impact these animals, forcing us to rethink resilience in the wild and, well, our own connection to these ecosystems.
That's precisely it. Our mission today is to extract the most important insights from our source material, to truly understand the different types of fungal infections, how they manifest, and what makes these distinct animal families vulnerable. As we explore, you're going to discover some surprising commonalities between camelids and cervids, but also some critical, often unique differences in how these diseases affect them. It's quite revealing.
And to do that, we're going to be exploring everything from the specific fungi involved and how these animals get infected, to what symptoms veterinarians and wildlife managers are looking for, how these diseases are diagnosed and treated. Crucially, we'll also delve into why this topic extends far beyond just animal health. Okay, let's unpack this. To make sense of it all, let's start by categorizing these infections, much like our sources do, which provides a really clear framework: superficial, subcutaneous, and systemic. Beginning with the superficial, the kind that affects the skin and hair, we find wingworm, also known as dermatophytosis. It's an incredibly common infection across both Camelidae and Cervidae. Seems like everyone gets it.
Indeed, very common. The primary culprits here are typically Trichophyton spp and Microsporum spp. What's particularly noteworthy, especially for cervids, is that Trichophyton varicosum stands out as a key pathogen often identified in those populations. That one keeps popping up.
For something as common as ringworm, how did these large animals actually contract it? Is it just direct contact, or are there environmental factors at play making it so prevalent?
That's a great question. Our sources make it clear that while direct contact with an infected animal is definitely a major route, there's also significant transmission through contaminated equipment or the environment itself, like shared fence posts or bedding. What's critical to remember is that those fungal spores are incredibly resistant. They can hang around in the environment for a long, long time just waiting for a host. They're truly tenacious.
Wow, okay, so hard to get rid of. And what clinical signs should people be looking for? What does it actually look like?
Well, the clinical signs often give it away. In camelids, you're typically looking for circular alopecia patches. That's hair loss, often with crusts, scaling, and sometimes itching. Pretty classic look. For cervids, the lesions are similar: hair loss, crusting, and scaling, but with notable redness or erythema. Usually easier to spot that redness. These lesions usually appear on the face, neck, and limbs. Common spots.
This is where we see a really profound example of the One Health concept, isn't it? The research highlights that the epidemiology of Trichophyton varicosum specifically points to domestic cattle as a primary reservoir. So this isn't just about animal health. It poses a direct occupational risk for humans in contact with co-grazing wild cervids, farmers, vets.
Absolutely. It genuinely blurs the lines we often draw between wild and domestic health, making this a critical interface for disease surveillance and human safety, a real crossover point.
So how do vets figure it out for sure, and what do they do?
For diagnosis, veterinarians rely on clinical signs, sometimes using a Wood's lamp for certain microsporum species, though that's not always definitive. Fungal culture and PCR are really the most definitive methods. Get a sample, grow it, or test the DNA. Treatment generally involves topical antifungals, like miconazole or clotrimazole, creams or washes. In more severe cases, systemic antifungals might be necessary, pills or injections. But critically, successful treatment also requires essential environmental decontamination because, as you mentioned, those spores are tough. You have to clean everything.
Right, otherwise they just get reinfected.
Exactly. Prognosis is usually good, but without proper decontamination, reinfection is a real risk. It can be a frustrating cycle.
Okay. From skin deep, we dive a layer deeper now into subcutaneous infections, getting under the skin. Here, the focus is squarely on pitheosis, an infection that affects both camelids and cervids.
The causative agent for pitheosis is Pythium insidiosum. It's a fascinating organism because it's not actually a true fungus. It's more like a water mold-like pathogen, an oomycete technically, and it thrives in wet, warm environments. Think swamps, standing water. Transmission typically occurs when open wounds or abrasions are exposed to contaminated water or soil, so it's very much tied to their environment where they're wading or grazing.
And what do these infections look like once they take hold? Sounds nasty.
They can be. The research describes the clinical signs as ulcerative lesions with necrosis, that's tissue death, and excessive granulation tissue, sort of proud flesh. These are often found on the limbs or abdomen. What's particularly notable for camelids is that the infection can also involve lymphatic vessels, leading to lymphedema, which is a significant, often disfiguring swelling. Really problematic.
You know, for something like pitheosis, where surgical excision is often the preferred treatment, I can't help but wonder about the practicality of that for wild cervids or even large, sometimes less cooperative camelids. Do our sources address those real-world logistical hurdles in treatment? It sounds difficult.
That's a very practical question, and yes, the sources do acknowledge the challenges. It's not easy. Diagnosis can be difficult, but typically involves histopathology, looking at tissue samples under a microscope, or PCR, serology, or a culture of the affected tissue. As for treatment, surgical excision of the lesions is often the preferred route if possible, yes, trying to cut out all the infected tissue. This is often combined with systemic antifungals like itraconazole or terbinafine. Interestingly, for camelids specifically, immunotherapy is also mentioned as a treatment option, trying to boost the animal's own immune response.
So multiple approaches needed.
Right. The prognosis for pitheosis is generally guarded, highlighting that early diagnosis and aggressive treatment are absolutely crucial for improving outcomes, especially given those logistical hurdles you mentioned. Catching it late makes it much harder.
Moving deeper now, we arrive at systemic mycoses. These are internal infections, and they are often far more severe, truly life-threatening. This is also where we truly see the vulnerabilities of camelids and cervids begin to diverge significantly based on the sources.
Absolutely. We'll begin with a critical and often fatal threat to New World camelids, coccidioidomycosis, also known as valley fever in humans.
Ah, valley fever. And this is where we really see that divergence we talked about earlier.
Yeah.
Unlike cervids, this disease, caused by the soil-dwelling coccidioids SPP, is a danger not prominently documented in them. It seems to be a very specific and severe vulnerability for camelids, llamas, alpacas especially.
Exactly. The sources emphasize the extreme susceptibility of camelids to this infection. They just seem particularly prone to getting very sick from it. And what makes it even more challenging is that pharmacokinetic issues, how the body processes drugs, make standard oral antifungal therapies largely ineffective in them.
So the usual pills just don't work well.
That's a significant hurdle because it means their bodies struggle to process and absorb the drugs effectively. It's incredibly difficult to get the right concentration where it's needed to fight the infection. It's a real treatment nightmare.
Turning our attention to aspergillosis, it's another opportunistic infection that impacts both groups, though our sources particularly highlight its presence and severity in camelids.
Yes, Aspergillus. The causative agents are Aspergillus spp, with Aspergillus fumigatus being especially prominent, very common mold in the environment. Transmission occurs through the inhalation of spores, often from contaminated hay, bedding, or dust, just breathing it in. It's crucial to note that immunocompromised animals are at a significantly higher risk for developing this infection. It really preys on weakened immune systems, stress, other illnesses.
Clinically, what would you observe in an animal with aspergillosis? What are the signs?
You'd typically be looking for respiratory issues like nasal discharge, difficulty breathing, maybe some coughing, general unthrifty signs too. Other common signs include weight loss and anorexia, just not doing well. In severe cases, it can become a disseminated disease, meaning it spreads and affects multiple organs throughout the body, like the lungs, kidneys, even the brain. That makes prognosis much poorer.
And how is it diagnosed and managed? Sounds tough.
Diagnosis often involves radiography, x-rays, and endoscopy, looking inside the airways, plus fungal culture, serology or PCR, from respiratory secretions or affected tissues, trying to confirm the fungus is there. Treatment relies on systemic antifungal therapy, maybe itraconazole or voriconazole, combined with supportive care, trying to help the animal fight it off. However, the prognosis is guarded to poor, especially if the infection is disseminated throughout the animal system. Once it spreads, it's very hard to control.
And we also need to mention cryptococcosis as another significant systemic infection identified in camelids in our sources.
Yes, that's caused by Cryptococcus neoformans or Cryptococcus gattii, another one you inhale. Transmission is through the inhalation of spores from contaminated environments, often associated with bird droppings, especially pigeon droppings. Clinical signs can include respiratory issues, weight loss, anorexia, and in severe cases, even central nervous system involvement. Neurological signs are a bad sign. Okay, now shifting our focus to systemic infections that are more prominently documented in Cervidae, let's talk about histoplasmosis.
Right, histoplasmosis. This one is caused by Histoplasma capsulatum. And the transmission route sounds familiar. Inhalation of spores from contaminated soil, which is often associated with bird or bat droppings. Similar pattern.
That's right. Common theme there with disturbed soil and droppings. Clinically, Cervids may present with respiratory signs like coughing and dyspnea, difficulty breathing, along with weight loss and lethargy. Again, those general signs of illness. The infection can also disseminate to other organs. The prognosis is guarded to poor if the disease is disseminated, but, as with most of these, it improves significantly with early diagnosis and intervention. Catching it early is key.
To conclude our overview of systemic infections, let's touch on candidiasis in cervids. That's caused by a yeast, right?
Exactly, Candida albicans. And it's frequently an opportunistic infection. This means it typically strikes animals that are immunocompromised or otherwise stressed, maybe on long-term antibiotics. It takes advantage of a weakened state. Clinical signs can range from mucocutaneous lesions like in the mouth or on skin folds and gastrointestinal issues to, in severe cases, systemic involvement. Prognosis is generally guarded, especially when it becomes systemic. If it stays localized, maybe better, but systemic candida is serious.
So circling back to that concept of opportunistic mycosis, like aspergillosis and candidiasis, it's really important to remember that their presence often serves as a sentinel. They're like an alarm bell, aren't they? Indicating underlying host health issues or maybe significant environmental contamination with fungal spores in both of these animal families tells you something else might be wrong.
Precisely. And this brings us to the bigger picture, one that truly underscores the One Health approach we talked about earlier. The critical importance of environmental management and preventive measures mentioned in our sources. It's not just about treating the animal.
This is where it gets really compelling, I think. The epidemiology of these diseases is not static, it's changing. Our sources highlight that the geographic ranges of environmentally sensitive fungi, like coccidioids and aspergillus, are predicted to expand due to climate change, warmer, maybe wetter conditions in some areas.
Right. That's a huge dynamic factor to consider in the future of these diseases. Absolutely. This has profound implications for both animal and public health, posing new and evolving threats that absolutely require proactive adjustment in surveillance and diagnostic capacity. We need to be ready.
It's a call to action for the scientific community, reminding us that health landscapes are always changing. They're not fixed. Our sources even detail specific strategic recommendations that connect animal health directly to human well-being. For example, they recommend monitoring Trichophyton verrucosum in wild cervid populations, especially at the livestock-wildlife interface, to track this zoonotic pathogen, meaning one that can jump from animals to humans. Keep an eye on where farms meet forests.
They also suggest targeted public health campaigns to increase awareness among agricultural workers, veterinarians, and rural communities about the risks of transmission from animals and the importance of hygiene and early medical consultation. Simple things like washing hands, wearing gloves, seeking help if you see a suspicious lesion. It's about empowering people with knowledge.
And this next point is fascinating, the concept of utilizing sentinel species, using animals as early warnings. Specifically, domestic canids, dogs, and camelids can be used for monitoring the environmental presence and risk of coccidioids to inform human public health alerts. Their infection rates literally become a warning sign for us, a biological barometer for the environment. If dogs and llamas are getting sick, maybe people are next.
It's a clever approach. Finally, our sources reinforce the need for strict biosafety protocols for all personnel conducting necropsies or laboratory cultures on animals suspected of having coccidioidomycosis. This is crucial for preventing occupational infections and protecting those on the front lines, ensuring the people studying these diseases don't become victims themselves. Lab safety is paramount.
As we bring this deep dive to a close, we've journeyed through the intricate world of fungal diseases affecting camelids and cervids. Quite a tour. We've explored the spectrum from common, though persistent, skin infections like ringworm to the far more severe, often life-threatening systemic conditions. We noted unique challenges like coccidioidomycosis in camelids and the shared One Health implications of ringworm for both animals and humans. It's been a truly eye-opening exploration into a side of animal health that often goes unappreciated. You don't always think fungi first.
No, you don't. The key takeaways for you should be a clear understanding of the different categories of fungal diseases, their varied transmission routes, and the critical role that environmental factors and host immunity play in determining vulnerability and disease progression. It's a complex interplay: environment, host, pathogen.
So this leaves us with something to think about. This raises an important question. As our climate changes and human-animal interfaces become more complex, how might we proactively adapt our strategies to mitigate the spread of these evolving fungal diseases, ensuring the health of both wildlife and human communities?